I'm Nina Krasinski, and I'm the Grant Science Support Coordinator for NOAA Ocean Exploration, where I support Dr. Laura Elmore and our partners at the Ocean Exploration Cooperative Institute. Thank you for joining us for today's NOAA Science Seminar. This is the fourth and final webinar of this four-part series about NOAA's Ocean Exploration Cooperative Institute, or OECI. During the first three webinars, we met the OECI team and covered a wide range of topics, from data to multi-vehicle operations and to education and outreach. Today, we will continue to hear more about how we conduct our deep sea exploration projects. Throughout the seminar, the speakers will discuss the various projects that were advanced during a recent OECI expedition across American Samoa. For your awareness, this presentation is being recorded and will be posted with closed captioning on the site shortly after the webinar. You will be able to ask questions following this presentation in the live YouTube chat box, but you must be signed into a YouTube account to do so. Otherwise, please email your questions to oeci@etal.uri.edu. So today, you'll be hearing opening remarks from Dr. Adam Soule, followed by presentations from Val Brown, Dr. Chris Sherman, Dr. Mary Burkitt-Gray, and Dr. Santiago Herrera. So it is now my pleasure to introduce Dr. Adam Soule, the Ocean Exploration Cooperative Institute Director, to kick off the webinar. Over to you, Adam.
Thanks so much, Nina, and thank you to all the folks online who are tuning in. I'm really excited for this seminar today because it puts a nice capstone on what we've talked about through the previous three seminars. This particular expedition that we're going to talk about brings together the technology development, the complex operations, the diversity of science involved in ocean exploration, and the education that the Cooperative Institute strives to support. So the expedition we're talking about is in American Samoa, the most remote U.S. territory from the mainland of the United States. This area hosts an incredible diversity in its marine environments and its biology. In the month of September, the Ocean Exploration Cooperative Institute executed an amazing expedition to explore this area. The expedition brought together a tremendous group of scientists from across the OECI and beyond that have interests ranging from geology to hydrothermal fluid flow, ecosystem function, marine resources, marine archaeology, biodiversity, and technology. Those folks were on the ship and on shore participating remotely in this expedition. Likewise, the expedition included a range of sponsors, most notably the NOAA's Office of Ocean Exploration, but with additional support from the NOAA Office of Marine and Aviation Operations, in particular the Uncrewed Systems Operations Office, the Bureau of Ocean Energy Management, and in-kind support from the National Marine Sanctuary of American Samoa. I won't go on in too much detail about the science because we have a tremendous lineup of scientists who are going to share their experiences from the expedition. So with that, I will introduce Val Brown, Research Coordinator of the National Marine Sanctuary of American Samoa, who's going to give us an overview of the sanctuary priorities and the observations that we were able to make on this expedition. Off to you, Val.
Talofa. Thanks, Adam, and talofa, everyone. I'm really happy to be here talking with all of you today about the recent Emamana o Natafale expedition. If you would have asked me as a young person living in Indiana, dreaming of faraway coral reefs and while I was studying lakes and songbirds, I would have never thought of myself being a member of a deep sea expedition. And yet, this past September, I found myself aboard EV Nautilus for the Emamana o Natafale expedition. And it took a while to get here. As Adam, if any of you have listened to some of our video, you'll hear me get teased about fish and coral reefs a lot. And that's because I really did make it to those coral reefs. I dreamed of that for a long time. My focus for 20 years has been on coral reef management and conservation. And for most of that time, I worked in Guam as a coral reef fisheries management liaison for NOAA's Coral Reef Conservation Program, and worked on a wide range of projects, including coral reef monitoring, rapid response, restoration, and citizen science. But in 2019, I became the research coordinator for the National Marine Sanctuary of American Samoa. And this is one of 17 sanctuaries across the country. It's currently the largest of those national marine sanctuaries in the United States, and it is the only one south of the equator, way down there in the bottom of the map. This sanctuary includes amazing coral reefs, which is part of what brought me here, like Fungatelli Bay, and the Valley of the Giants in Tau, which is home to the astounding and iconic Big Mama, one of the largest and oldest shallow water corals in the world. And some of these remarkable reef areas here have been studied since the 1980s. And this sanctuary is really interesting in the fact that it is tasked with incorporating traditional knowledge and stewardship into the management of our resources. So Fa Samoa, the traditional Samoan way of life, provides the cultural context for all sanctuary activities and functions. And our site has been a leader in ocean literacy and community engagement efforts in the territory, providing professional development for teachers and a range of activities for students and community members, including our Science on a Sphere. Today, National Marine Sanctuary of American Samoa includes 6 units across 5 islands. We're spread across the American Samoa Exclusive Economic Zone, and it includes over 13,500 square miles of open ocean and deep sea. Now, while the coral reefs have been studied since the 1980s, these open ocean and deep sea areas have been poorly studied, so we don't know very much about what lies below the surface. Prior to this expedition, there had only been 3 expeditions with submersibles or remotely operated vehicles, and really limited work in the upper layers, mostly linked to commercial fisheries. As we worked with experts to evaluate data for our 2020 Sanctuary Condition Report, it became clear that we did not know enough about these habitats. So at the time of that publication, there had only been 15 submersible and 28 ROV dives across that entire 13,500 square mile area. And there were so many gaps in our knowledge about these vast ecosystems that we were challenged to even evaluate their status. And so we were really thrilled when OECI, the Ocean Exploration Cooperative Institute, and Ocean Exploration Trust were willing to put together another expedition for American Samoa. And not only did they bring the ship, but they brought an astounding array of tools and expertise with them, and also held space for our educators and for interns as well. To kick off this effort, the sanctuary worked with Ocean Exploration Trust to host a deep sea workshop with local partners and community members back in March. And this was really an important step in engaging the local community in this effort, helped us identify community priorities for research and outreach that included things like understanding where bottom fish might be residing, just even what's down there on the seafloor, and maybe even providing some information to help people think about the risk of deep sea mining and whether that's something that the territory wants to pursue. This workshop was followed by a professional development for educators that was supported by Ocean Exploration Trust, NOAA Ocean Exploration, and the Cooperative Institute in August, which helped encourage support for this effort, get all of our educators excited and brought on. And so our local educators, Isabel Halituatuia from the Sanctuary, a local teacher, Setefano Umanga, and Maggie Offa from the Department of Education, all sailed on the Nautilus during this expedition and provided communication support, including ROV telepresence and classroom presentations throughout the cruise. That team was able to host 102 ship-to-shore interactions during the expedition, and they reached over 3,000 learners in 30 U.S. states, 4 territories, as well as some foreign countries. And in addition to that, The interns that were on board, I think they were just shown on screen a second ago, graciously provided presentations to local schools after the cruise. So when they could have been running around or relaxing, they actually came out to our schools and provided information and encouragement for our local students to pursue marine careers, which was really fabulous. But let's get to kind of more of the science and the data. The data collected by this expedition is going to address some really important priorities for resource assessments and characterization, and research needed for the sanctuary to help us guide our sanctuary management. Um, the ROVs, Hercules and Atalanta, as well as AUV Sentry, really allowed us to peer into the depths and explore new sites, and sites like the Wailuulu Seamount that have been previously studied but are dynamic and changing, and that we wanted to revisit and understand better. And so there were 17 successful ROV dives totaling 187 hours of seafloor exploration, 10 Sentry dives, and 142 hours of exploration with it. And over 508 samples were collected. And so this is a huge amount of information that's going to help us manage these areas better and inform our community about what is beneath the surface of the ocean. And so the expedition, one of the things we were most excited about is some of those samples, actually the bulk of them, I think, are environmental DNA samples. So those are samples of the little bits of DNA that are floating in the water, maybe from fish scales or feces or little bits of all of the critters that are down there. And these samples are then going to be processed and matched to known species. Is really going to help us understand the biodiversity of our open ocean and deep sea areas in ways that we've never been able to do before. And so having the eDNA auto samplers that were able to filter large amounts of water, and those were mounted on autonomous underwater vehicles like Mesabot, the Deep Autonomous Profiler lander, and support from the autonomous surface vehicle DRIX. All of that brought together just really took this expedition to another level. And we're so thankful to Ocean Exploration Cooperative Institute for bringing all of that together, putting together such an amazing team. We had researchers from Woods Hole, from University of New Hampshire, University of Rhode Island, and Lehigh University that we had never met before and worked with. And now we have this collaboration with all of these experts, which I think is going to take the management of the sanctuary to a whole nother level, down to that deep, deep seafloor. But as you can imagine, there's a lot of data to be collected during this expedition, so it's going to take some time to analyze it. And you're going to hear about some of that amazing work from our other speakers. And hopefully within the next few years, we're going to be able to come back and talk about the results of all of this and start putting some of it together and talk about how we're actually using it to improve sanctuary management. And with that, I'm going to hand it back to Adam.
Thanks, Val. That was tremendous. And I echo everything you said, and just would mention how gratifying and exciting it was to learn about the Samoan culture while we were there in American Samoa and interact with the people. So that is much appreciated. Next, we're going to hear from Dr. Chris German, Senior Scientist at the Woods Hole Oceanographic Institution, who was the lead for hydrothermal plume hunting activities that we undertook. So over to you, Chris.
Thank you, Adam. So what I was doing on the ship was, well, I was participating in the ROV dives with everybody else, of course, which was the main activity on the ship. But at the same time, what I was also doing was working with a second vehicle that we brought along called the Sentry Autonomous Underwater Vehicle. And the reason that we brought this along as well is that this is a free swimming robot, so it can act in parallel with the ROV. We can have both things in the water at the same time. And what I was doing was using the vehicle to try and help explore what was happening in particular around the Wailuulu Seamount, to see if we could actually find additional hydrothermal sources that can actually increase the efficiency with which we explore with the ROV. So finding new targets even while the ROV is doing its thing. And the way we use this vehicle is shown in this artistic illustration here, that a hydrothermal vent on the seafloor is full of hot water, if you get the right kind of system, it has a stem of warm water that rises up into the water column. It kind of dilutes as it rises, and then it flows out laterally through the water column. So if you have a vehicle flying backwards and forwards, as shown in this along the blue dotted lines, if Sentry is doing that kind of thing, it can actually map out where the plume is, work out when it's got close to the center of that plume, and then that can actually help target what to do. In this illustration, it was just lowering a device for collecting water samples, but in our case, it was more a case of could we actually use this to help find new places to dive with the ROV beyond vent sites that we already knew about. On this particular cruise, though, the main thing we were really doing was developing some new technologies to try and improve how we actually use the vehicle like that. So we actually started by going to a vent site that we already knew about around the rim of the crater of the Valerie seamount, which actually had an active vent with gas bubbles rising up from it. So you can see this towards the center of the picture here. You can see this brown mound with a white kind of bullseye at the center of it. And as you come into close to it, you can see there's about a 1.5 meter tall, so that's about 4 feet tall, chimney with gas bubbles coming out of the top of it. And so this plume of gas bubbles rises up above the seafloor and then it spreads out sideways. And so that was the plume that we decided we were going to go and actually use, so that when we put Sentry in the water, we could have it flying around at the depth that those bubbles ponded out at, and use that with various different chemical sensors that we'll have Mary talking about that coming up soon, to actually map out where the source might be. So this is really at the very cutting edge of how we do our exploration. So to try and explain that, in an autonomous vehicle, normally you drop it over the side of the ship, it flies around, it collects whatever information it's going to collect during a survey. So it's got a preloaded mission of what it should do. And traditionally, the vehicle just goes off and does that. And there's nothing you can do about it. And you don't actually know what it's measuring until you bring the vehicle back at the end of its dive, maybe a day or more later. Then you bring it back on the ship, you plug into a connection, you actually download all the data from the robot onto a computer, and then you begin to analyze it. What we've actually been doing in the last just over a year is to actually try and streamline that. And so what we started doing last year for the first time was that we actually had a way to use acoustic communications that we can actually smuggle little bits of information from the robot up to the ship via our navigation method. So using sound waves, taking little packets of information and sending highlights of what the vehicle has found so far about once every minute or so that people on the ship could actually see that. And in fact, here we've got the photograph that the gentleman in the blue jumper is Guangyu Zhu. So he was actually the chief scientist for the program that we were doing here. And sitting next to him is another one of our colleagues, Victoria Preston. So Mary and I were actually out at sea as part of a team of 6 people that were doing that last year. And that worked really successfully in terms of getting the data from the robot to people on the ship, but no further. What we were doing this year, which was a new thing, was to actually push the technology even further. So what we actually did was took the team of 6 people who were on our cruise last year, who were all working together at sea. And now what we did was we actually kept me and Mary out at sea because we needed to be there. But then all of Victoria, Guangyu, and Andrew, who are shown in the right-hand side here, were all at the 3 different locations in the US, in different places, in different time zones. So Victoria, who actually runs the software that enabled a lot of this, the communications, was in Boston. Guangyu, who's the overall project lead, was in Seattle, and Andrew was in Denver. He works for NASA's Jet Propulsion Laboratory, and he's the person we're going to talk about most, while Mary and I were over in American Samoa, on top of the seamount. And so what we were doing now was not just bringing the data from the robot to the ship in real time, but because we were on board the EV Nautilus, which has this ability to telemeter back to shore and has very high bandwidth, we were sending all the data onwards to Andrew in Denver. And the reason we were doing that was that he's been developing a new software program to see if he could actually train a robot to search for vent signals automatically, and basically put me out of a job. So my career has always been to look at data from the Sentry and actually work out where hydrothermal anomalies can be. And so in this map, it'll show you what happened. In the outer blue box was we sent the vehicle down and said, go and do a broad survey that's generally in the plume. So it was actually starting near the vents that we knew about. And then during the course of that plume survey, what it was doing was using its sensors to work out, okay, which corner of this box was the most promising? Where were the places where we saw the strongest plume signals? And then at the end of the mission, what we were doing was sending the data, and instead of me making the decision about where the most interesting signals were, what we were doing was asking Andrew's computer program, without Andrew doing anything other than feeding the data into the computer program, to decide where we should go. And then Andrew's program then actually said, this is where the vehicle should go, sent that information, squirted it back out on Andrew's computer. He relayed that to me. I made sure we weren't going to put the vehicle in any difficult place. I told the Sentry team, and then they were actually able to make the vehicle go and dive in that area. And so in both of these two pictures, and this one is the second time we did it, what we actually found was the little red box is actually where the computer program in Denver told us Sentry should go. And to cut to the chase, the little red circles in each of those boxes in the maps shows you where the strongest anomalies were. So basically the program worked exactly right. It sent us to exactly where we should expect, or where I would have chosen to send the vehicle to. So now we have this whole thing completely automated, which is a great step forward for the future. What it also did in the second case, though, was in the first case, we found anomalies right over the vents that we already knew about, and that was kind of what we expected. What was a bonus was in that same survey, there was a hint that there were additional anomaly signals further east and up over the top of the rim of the crater and down on the outer flanks. And so that was really valuable, because then when we were interested in, well, having learned about that, then when Val, who you just heard from, said, well, you know, the sanctuary would also be interested in looking at the outside of the volcano, as well as on the inside and around the rim for the hydrothermal sites, but just looking at the ordinary biology, we knew that we should also be on the lookout for some kind of lower temperature venting on the outer flank as well. And on the very last dive at the seamount, at the very top of this rim, as we came up towards that same area that Sentry had indicated, we did indeed actually get to see some additional low temperature venting sites, which I think we probably don't have photographs just yet, because we either saved them. Oh, there's a few photographs here. We actually spotted them, first of all, from some very bright pink corals that were in this area. So there's just a very high abundance, but of low diversity, just a single coral type. But what we could also see in the top left picture were these very pale yellow patches, and that's where they were microbial mat. And when we went and zoomed into those brightest yellow color areas, that's where we found exactly what Sentry had been hinting at, was this kind of lower temperature flow that was not inside the crater that was known about before, not around the rim of the crater that we knew about before, but actually on the outside as well. And I think we'll hear from Mary later on about how she was actually then able to use the same chemical sensors that she'd used on Sentry to help us find things, to then go and actually study some of those fluids as well. So that's my part of the story. And Adam, now I'll hand it back to you to get on with what comes next.
Thanks, Chris. It was truly amazing watching the Sentry vehicle work, and having used it before, these new tools are going to make it much more efficient and effective for some of the operations it does, in particular finding these anomalies. So really appreciate you being able to participate in this way.
Yeah.
Our next presentation comes from Mary Burkitt-Gray, a research associate at the Woods-Hole Oceanographic Institution, who was out on the ship applying some new in-situ chemical sensors, and I'll let Mary tell you about it.
Hi, my name is Mary Burkitt-Gray. I'm a researcher at Woods-Hole Oceanographic Institution, and it's really great to follow on from Chris, who was talking about this, the big picture, how we're going to map this whole hydrothermal area, because I'm going to follow on and talk about the development of some new oceanographic sensors and how we use those on this research expedition. So there's a lot of really exciting opportunities at the moment to develop new technologies that measure chemical or physical properties of the ocean. This field's had a lot of really rapid advancements recently, because there's a lot of new optical components that are becoming much more widely available, much more affordable, such as new light sources like LEDs and new fiber optics. And these have really advanced very rapidly in the last decade, in part because of some of the advancements of the telecoms industry that's driven forward this development. So it's really exciting to be able to then take these new technologies and apply them for ocean research, and take all these advancements to be able to uncover new properties of the ocean that we haven't really been able to study before on this spatial or temporal scale. So during this expedition to American Samoa, we're really excited by the opportunity to deploy a new sensor which detects the dissolved gases in the ocean. So this sensor has been developed here in the Chemical Sensors Lab at Woods-Hole Oceanographic Institution. And that development's been led by Jason Caput and Anna Michelle. So this sensor is called SAGE. And I think we have a diagram of how we install it on a vehicle, for example. So the development of SAGE was actually originally funded by a NOAA Ocean Exploration Award. We can install it on vehicles or such as ROVs or AUVs. And just as a brief overview of how it works, so we have seawater that's pumped continuously in front of a membrane. And behind that membrane is a lower pressure environment. So that entire housing is actually under a vacuum. And the space behind that membrane is then connected via a very, very, very small hollow optic fiber to this vacuum. So that allows us to draw gases out of the water and then through this entire optical system. And then we use a technique called absorption spectroscopy to measure the amounts of various gases that we have extracted out of the seawater. So on this research expedition, we were particularly interested in looking at carbon dioxide and at methane. This technique is really exciting because previously, when scientists have wanted to study the amount of gases such as methane or CO2 that are present in seawater, we've primarily had to do this by collecting individual bottled samples of the water and then bringing those bottles back to the ship or back to the shore to run various analysis on them there. So, for example, we might use chromatographic methods to study the composition of different gases that are dissolved. But we're pretty limited with some of these approaches. You have to be quite selective about where you acquire samples because you only have a limited number of bottles that you can acquire on a dive or that you can collect on a CTD rosette. And it's also hard to characterize how this composition changes through the process of sampling. So, for example, how much degassing you get during the process of bringing it to the surface and running the analysis, or whether you have any biological processes that are continuing to go on in your bottles, and that can skew your in situ gas results. And one other problem that we've had before is you don't get any prior information about what gas is present in the water when you're choosing where to collect a bottle sample. So you might use a salinity sensor, a pH sensor, a temperature sensor to indicate that there's something going on, but it won't be until hours or days or weeks later that you're actually able to find out what the gas composition was and what was present. This can sometimes also lead to some interesting selection biases. So, for example, if you're on an ROV dive, you might choose to take samples for gas analysis based on the ecosystem you're observing. So you might see some very interesting microbial mats, for example, and you have the sort of the concept that you think that's going to have some interesting gas properties, but then that might also lead to you missing really scientifically interesting measurements or interesting observations because you didn't have any sort of prior observations about what those ecosystems would look like. So this is one of the really exciting advancements we've had with deploying SAGE in this expedition. So we're able to conduct a lot of research in the volcanic regions in the National Marine Sanctuary, so at the Valerie Seamount. So these volcanic systems discharge a lot of carbon dioxide of magmatic origin. And we've been able to get some real-time information about these gases that are being discharged from these volcanic regions. We've been able to track them all the way through the water column, and we're hoping to be able to study the fate of these gases as they pass up from this hydrothermal origin through these plumes. So as Chris mentioned earlier, we've been able to look at the gas concentrations in real time. So in the ROV van or in the Sentry lab, to be able to study how these concentrations are changing and what their dynamic processes are, and we're able to tell that in real time during the dive. So that's been a very exciting advancement, and that's probably one of the first times that there's been really good in situ real-time CO2 observations in this seamount. So we used two different ways of deploying SAGE during this expedition. So one of them was through a deployment on AUV Sentry. So as Chris mentioned, this is an autonomous vehicle, and it allowed us to acquire continuous CO2 measurements throughout the entire deployment and stream these back to the ship to get updated information on the relative location of Sentry and the hydrothermal plume. So SAGE was installed on the bottom of the vehicle, as you can see in this photo here. And the inlet was routed to be really close to some of the other sensors that we often use for hydrothermal studies. So, for example, there's a MAPPA sensor that looks at the oxidative reductive potential of the seawater, and it also looks at the turbidity. So that's the amount of particulate or poorly dissolved matter that scatters light back. So this is allowing us to draw together these two different data sets of our long-term understanding of hydrothermal systems, and now we can bring that into the dissolved carbon dioxide. One thing that we're really hoping to be able to get out for this is how much carbon dioxide is then released from these volcanic sites and what is its fate. So Sentry surveys are a really great tool for this because we're able to map the hydrothermal plumes at various different spatial resolutions. We're able to look at various different depths, and we're able to be responsive in the dive based on the observations we're getting from SAGE about where the highest concentrations of carbon dioxide are located. And we're also really excited to be able to combine this with the other data that we're getting to be able to estimate the total amount of carbon dioxide that is being released from these vent sites, and then what the impacts are on the surrounding oceanography, what the impacts are on the CO2 distribution, on the pH, for example, and what the depths that accumulates at, or where that goes. So the overall lateral spread, the buoyancies, and then the chemical properties that changes as a basis of these CO2 emissions. We were then also able to use the results from these Sentry surveys to inform where we would want to dive with ROV Hercules. So in the previous dive map, you could see that there's this hotspot at the venting site, and we're able to directly trace that hotspot back from a 30-meter altitude to the origin of the hydrothermal vent, which was this really cool bubble plume, bubble origin that Chris also mentioned in his talk. So this was really interesting because we had a really wide range of biological and geological features in a very small spatial area. So for example, we had very high carbon dioxide in the vicinity of this bubbling vent, but then we actually had sort of equally high carbon dioxide measurements that were very close, but were associated with very different geology and biology. So for example, we detected very high CO2 in the regions of these iron oxide or oxyhydrite-rich chimneys, but the actual flow that was visible from these chimneys was much more diffuse, was much lower temperature, and without any visible bubbling. And I think, yeah, exactly. So we have this very different geology and very different fluid properties compared to the venting site that was really only, tens of meters apart and at the same depth. So we're hoping to be able to understand from these SAGE measurements much more about the geological process in this region that result in these very different dynamics with very little spatial separation, and how these different flow properties result in different fates of the carbon dioxide in the water column, and how they impact the overall chemical and biological processes that are also happening. One really exciting thing about having the SAGE sensor on the vehicles was we're also able to sample from very, very small spatial regions. So for example, we're able to use this wand we called the drinking bird that basically has a hinged sampler that we're able to drop down to take specific water samples right from where we saw a venting site. And this is really great for them being able to combine that data with temperature data, for example, so we can really understand the chemical and the temperature, the thermodynamic properties that are happening in those regions. This is also really great for then combining this data set with more sort of long-standing gas analysis techniques. So for example, we also collected some IGTs, these gas tight samples during the cruise of these same venting sites. And so by having this really sort of small spatial responsiveness of the SAGE sensor, by pumping fluid directly from these vent sites to the instrument, we're hopefully able to really capture a full dynamic spread of what's happening in the gas emissions. And one extra bonus we had from this expedition was that we were also able to acquire really high resolution data just about the CO2 distribution in the water column. So this is something that's actually quite hard to study normally because it does often involve taking bottle samples back to the ship. And so this was just a really great opportunity to study the different CO2 dynamics which are happening in the water column. So the surface layers, which are much more dominated by atmospheric mixing and respiration and photosynthesis, and down to the deeper circulation layers. And the deep Pacific is particularly interesting because it contains very high levels of CO2 compared to, for example, the Atlantic. And so it's just great to be able to acquire these really high resolution CO2 data sets that haven't really been studied in that region to much depth before. So with that, I will return you to Adam, and thank you for listening to some cool updates on CO2 data.
Thanks, Mary. That was fantastic. I will emphasize that sensors like Mary's describing that provide you real-time information about the environment in a new way for a new metric are so valuable for decision making when you're actually exploring an area. So an area that can look pretty run-of-the-mill might have tremendously anomalously high CO2 and kind of dictate further investigation of that area. So, not only is it providing new science, it's helping us make our observations and collect samples in a better way. So really appreciate it. All right, thanks, Mary. Our next and final presentation will be by Dr. Santiago Herrera, Associate Professor at Lehigh University, who was our lead for ecosystem studies in this environment. So over to you, Santiago.
Thanks, Adam. And I'm happy to be here. So I'm really excited to talk to you about some of the observations that we made in this expedition. And it goes really nicely after Mary's presentation. It brings back memories from when I started my career, thinking about the ocean as this very alien place. And that was a picture of me in the first field research trip that I did. 20 years later, we're talking about one of the most amazing expeditions that I've been able to participate in. And one of the really interesting observations that we're able to pair with the measurements of CO2 that Mary was describing with her fantastic instrument is correlating them with observations of biology. And so, for example, this is an image of the seafloor in the interior of the crater of Pelulu, the active seamount in American Samoa region. This volcano looked like this in 1999, and those images were representative of what the seafloor looked like there. It's full with CO2, but you can see in the last 20 years or so, there's been a development of a cone erupting or formed by eruptions in the center of the crater of the seamount. In 2017, we were able to visit this and take a look at what the fauna on different parts of the cone look like, and we found many different species, or actually just a handful of species, including anemones and hydrates, as well as sponges, carnivorous sponges living on the newest parts of the cone. And when we visited the older parts of the cone that were about 5 to 7 years older, we started to see corals and brittle stars. And I was really curious to go back 7 years after and see how things look like. And so this is images from what in 2017 was about 7 to 12 years old seafloor. Sorry, what it is now, 7 to 12 years old seafloor. So it was brand new seafloor in 2017. And now we're seeing those corals arriving in the scene. Those that were previously observed only in older parts. So we're seeing this succession series of animals coming in a particular order. And so we're really excited to also explore, to get the opportunity in this expedition to explore other parts of the seamount that are likely much older. And so from looking at things in the crater that look very much like this, we're able to go on the rim of the crater. And what we saw was this. So it's a much higher diversity of life, including multiple species like large shrimp and squat lobsters, different species of anemones, different species of sponges, and a very large abundance of these mushroom corals. And so these observations are giving us hints of what that succession process is over time, from moving from a cone that is not older than 20 years to areas around the crater or on the rim of the crater that look like this. We have a much higher diversity of animals, including the species that we see on the cone, but many different ones. In particular, we saw a lot of fish, including cartilaginous and bony fish that gave us some really nice displays of their behavior as we cruise along one of the edges of the volcano. Really exciting to be able to make these first characterizations of who lives at the different parts of the volcano, and we're really interested in correlating the age of the seafloor also with the chemical parameters that we're able to measure with SAGE and other sensors that we had on the ROV. Now, we also had the chance to dive outside of the volcano, and so this is some of the fauna that we saw in the outside. So we visited northeastern slope on the outside of the active volcano of Vailulu, where we saw, again, a bigger diversity of species. We also saw predators of anemones, including this sea spider, that are likely taking advantage of the large numbers of these animals that are found in Vailulu. On the outskirts of the volcano, we also observe very large corals, including bubblegum corals like this one with associated brittle stars and shrimp, as well as many other different kinds of deep sea corals, including golden corals, bamboo corals, black corals, cup corals. And then when we visited other volcanoes, other seamounts further away from Vailulu, they're inactive and are much older. We saw what those older communities can look like. And this is a great example of a diverse and abundant coral community and sponge community that we observe in other seamounts. So different kinds of species, right, reflecting a much more mature community. And so one of the great things about the expedition is that we're able to collect this information that allows us to understand how these communities, biological communities, are structured over time, but also space, and provide critical baseline information to the sanctuary and the region about the diversity that is found here, with the context of the age and the of the habitats and the environmental composition measured by the sensor, the array of sensors that we had on Hercules and Sentry. Some of these animals that we observe include some fantastic observations of things like this basket star that we found in a different seamount, inactive seamount in the region. These basket stars, like corals, are suspension feeders, so they use their bifurcating arms as a bit of a net when they spread them out to capture particles and likely zooplankton that they feed on. And so these diverse and rich communities, as I mentioned, give us the first insights about who lives there in the context of the diversity in a global context. The exploration of the seamounts also reveals some evidence of fascinating processes that occur as shallow seamounts, where the water column populations of plankton, they migrate from the surface to the mid-water twilight zone, can be found near the summit or at the seamount seafloor for seamounts like Vailulu that come close to the surface. And so this was another seamount where we observed, I think it's around 300 meters, we observed this fish, dragonfish, that were very likely migrating. And so this provided evidence that these animals that live in the water column can get close enough to the seamounts in the American Samoa region that are likely interacting with the fauna and the seafloor. And we saw evidence of this. We saw evidence of animals like this crab catching some of these organisms and feeding on them. And so one of the most exciting things that we learned through the expedition is that the volcanism that is happening at Vailulu and the hydrothermal output from the volcano into the water column is slightly modifying the composition of the water column enough that is enriching the productivity of the water above the seamount Vailulu, but likely other seamounts. And we see evidence of this in the high abundance of animals that are found on the seafloor of Vailulu. We saw some of these animals feeding on motile fauna, like these anemones that were feeding on crustaceans that were roaming around. Again, this hydrothermal venting and the nutrients that it brings are likely fueling biological communities in the water column that in turn are likely feeding the communities that we observed in the seafloor. But it wasn't only small animals that we observed taking advantage of this environment. We also observed large animals like this sunfish visiting the seafloor near the summits of seamounts region. I think in this particular dive, we observed 3 different organisms of sunfish at the depths that we were diving with Hercules. Again, providing evidence of this movement of animals from the near surface to the deep sea and back, transporting materials and influencing the flux of carbon and other important constituents of the water column in these ecosystems. We also observed large marine mammals, including whales, over the summit of Failulu and other seamounts, including some toothed whales as well as humpback whales. And so these are some footage that Samantha Ling Martinez, who was one of the video engineers on board, collected on this night that we observed this particular or really spectacular behaviors of humpback whales that visited the Nautilus as we were operating in some seamounts in the region. And so again, we were lucky to be able to make connections all the way from the seafloor to the water column and the surface using a diverse array of tools and expertise that was brought together to really succeed in achieving the goals that we set out on the expedition.
Thanks so much, Santiago. It's just amazing imagery of the organisms that are down there, and really important observations for what the ecosystems are doing. I just want to once again thank Val, Santiago, Chris, and Mary for their presentations today. We have some time for some questions from the audience. If you're signed into the YouTube account, you can ask questions in the YouTube chat box. And for those of you with .gov email addresses, in order to ask a question, you'll need to sign into YouTube with your personal email address, or you can email your questions to OECI at etal or etal.uri.edu. One of the questions that's come in, Mary, the CO2 emitted from the volcano, does that have an impact on climate change?
Hi. So one of the things is that right now, a lot of the dynamics of CO2 from these deep sea volcanoes are actually not that well studied or well understood. There's a lot of processes that contribute to the fate of that CO2, how there's a lot of different reactions that can happen with that, both from biological origin and just from carbonate chemistry in the seawater. So I don't think we're able to say yet what the atmospheric contribution is of CO2 from this volcanic system. Some shallower seamounts do seem to have a much more atmospheric contribution, but this one's pretty deep. So I don't think we can say from Valerie specifically whether we have an atmospheric contribution. It doesn't look like there's a very obvious one that we detected, but it's probably too early to say from the data we collected on this cruise. But I'm happy for other contributions from Adam or Chris or whatever, if you have opinions too.
Yeah, no, I think you're absolutely right. I think this is there's a lot of CO2 coming out, but not nearly to the scale that is being emitted by anthropogenic processes. So probably not a big lever on the system. Another question for Val: what do you view as the biggest threats to the marine environment around American Samoa?
Well, related to that last question, climate change. So in our 2020 condition report, climate change was cited as the biggest threat to all of our resources. And I think that's one of the things I didn't mention earlier, is that this expedition also can help us create kind of baseline to understand what is happening on the seafloor and in the water column in the open ocean and deep sea, to help us understand what the impacts of climate change are on those resources. We often think about the impacts on coral reefs, but the open ocean and deep sea areas can also be affected by some of the changes that we're seeing in the oceans. So important information to address that key threat.
Thanks, Val. Chris, a question for you. If we can now get a robot at the seafloor doing what a smart algorithm ashore tells it to, where can we take this next? Where might this take us next?
That's a really fascinating question, Adam. I think one of the things to think about is that what we were able to do on this cruise was very much testing whether or not the algorithms, whether the artificial intelligence is fit for purpose and is ready to start being used. So I think what we had was a very simple approach here of how the thing would work. But we're now perched at that point where, okay, the next step then is to take that same algorithm and cut out all the intervening factors. Why not just have the algorithm actually on board the robot so that it could actually be doing the processing itself instead of having this whole thing of going through several safety valves and back again? Now that we've actually proved that it's robust, then we can actually start giving these robots more opportunities just to like basically go off and do their own thing and then just tell us what they did later. And I think the reason that's important is we don't always have a ship and an AUV where the ROV is. As we get to more and more long range vehicles that could be left alone, if we can trust them to be left alone as well, then that's great. So the more we can actually build that intelligence into these robots is a fantastic thing. So that's why, as you were saying at the outset, it wasn't just NOAA's ocean exploration program that was interested in this, but also the uncrewed marine systems, part of the operational side of NOAA, is also really excited in this research because it allows us, instead of it being cutting edge like it is right now, it could be what becomes like standard practice in the future for letting larger fleets of robots to go out and help us with our exploration and monitoring the ocean a lot more effectively than we can do yet. That's really what I'm excited about.
Yeah, that's great. And I think what's exciting is that we can work on these technological developments for the future while also doing the science of today. One question I have that anyone can chime in on, what do you think was the most exciting finding or advancement accomplished on this expedition? And Chris just mentioned the advancement of this algorithm for plume hunting. For me, the ability to pair now what we're seeing in terms of hydrothermal processes and volcanic processes and biological processes with CO2 data is going to be a really big jump forward for the field. But I'm happy to open it up for anyone else who has their exciting advancement that they want to just share with the world, because they can't keep it in anymore.
I was just going to say they're really like, they're really comprehensive and interdisciplinary surveys that we did. So it wasn't just one specialty, it wasn't just one focus for this research, but it was everybody from lots of different fields bringing together their knowledge, and that I think is studied these regions in a way that hasn't really been the focus before, maybe.
Yeah, I strongly support that. That's what I would have said as well, is the interdisciplinary nature of this expedition, bringing together so many tools, so many experts. We also had the scientists ashore, and just being able to bring that kind of brain power and machine power and processing power all together was really exciting. And I think we're going to learn so much from it.
Yeah, I'll add that. Oh, go ahead, Chris.
Well, I think just the last one was then it was the fact that it wasn't just the principle of doing all these great things, but there were such great tangible results inside the lifetime of the cruise. That's what really got me, was because we had Mary's sensors, we could actually use Sentry to actually sniff in the ocean in new ways and sniff for new flavors of chemicals that we haven't been able to do before. And then from that, and the fact that we were doing it with Sentry, allowed us to make some predictions about, this is really kind of cool, and maybe there's some really new kinds of hydrothermal activity on this seamount, but we're not going to know about it because that's all we've got is an AUV. But instead, because Val was interested to go there anyway for the sanctuary, she was actually able to say, no, let's actually spend another dive on that volcano. And then during the course of that volcano, before the end of it, we actually found the kinds of vents that Sentry had been suggesting should be there because of Mary's data. So it wasn't just this is what we can do in the future. It's like, we did it like the very next day, pretty much. But we would not have been able to look for that stuff. We wouldn't have even known it was there to look for if we hadn't brought all this combined talent that we had. It was just one of the coolest cruises I've been on in such a long time.
One of the things that made it really special for me was the participation of the folks from American Samoa, Isabel and Stefano, to understand that the science we were doing is in their backyard and really exciting and important to them. Made it just that much more compelling to me to do the work, and so it was all in all wonderful. What a great way to wrap up. I'm going to pass it over to Nina to close us out, but I want to thank again all the presenters and the folks tuning in online, and the folks in American Samoa for welcoming us to do this science. Oh, I'm sorry. I'm supposed to hold on for one more question. And I'm holding. Yeah. So the question came in, 300 plus hours is a lot of video to watch. Is there a plan to extract the biological observations from this video? And does NMSAS, the American Sanctuary, have the capacity to do that? I'll let Val handle that first, but we can all share in on the answer.
Yes, that is a lot of data, a lot of video to process, and we actually don't have the expertise in-house to do that. So that's one of the great things about this partnership, is that we can recruit people that have the expertise to do it right and give us the best information from that process. And we're currently working on trying to line up funding and personnel to do that. So stay tuned. We do have annotations from the past expeditions, which we are compiling into a summary report that will hopefully come out in 2025.
Excellent. Yeah. So it should be noted that during our operations, we do a lot of annotation and identification of organisms as the dives are proceeding. So we have a first pass kind of collection of annotations. That is not to say that it's complete by any sense, but we had folks participating in the cruise and part of the Cooperative Institute who will go on to do additional annotation in collaboration with the sanctuary. And it becomes one of the data streams that comes out of this expedition that has a legacy beyond just the initial science that gets done. As Val said, a baseline for future observations. It's kind of a critical component of that. So I appreciate the question. All right, so now I will pass it back to Nina to wrap up, and thanks again to everyone.
Awesome. Thanks, Adam, and all the speakers for your wonderful presentations today. And also thank you all for joining us today and over the past several months as we learned more about the Ocean Exploration Cooperative Institute. As always, I'd like to thank all of our OECI partners and NOAA collaborators who make this work possible, especially the University of Rhode Island's Inner Space Center, who produced this entire webinar series. As I mentioned earlier, this is the final seminar of this 4-part seminar series. However, if you'd like to learn more about OECI, I would like to encourage you to check out the first 3 seminars, especially if you missed viewing them live. And you can do this by using the link that's on your screen. And yeah, I just wanted to thank everyone for joining us today and throughout the entire seminar series, and have a great rest of your day.